On July 5, 1996, a sheep named Dolly was born at the Roslin Institute in Scotland, marking a monumental achievement in the field of genetic science. Dolly was the first mammal to be cloned from an adult somatic cell, a scientific breakthrough that captured the world's attention and sparked debates about the ethics and future of cloning. This narrative explores the scientific journey that led to Dolly's creation, her life, and the profound impact her existence had on science, ethics, and society.
The concept of cloning had been a subject of fascination and speculation for decades, but it wasn't until the late 20th century that scientists began to make significant strides in the field. Cloning involves creating a genetically identical copy of an organism, and before Dolly, scientists had successfully cloned plants and simple organisms like frogs but not mammals. The breakthrough came at the Roslin Institute, where scientists Ian Wilmot, Keith Campbell, and their team embarked on a bold experiment. Their goal was to clone a mammal using a technique known as somatic cell nuclear transfer, SCNT. This process involved transferring the nucleus from an adult somatic cell, in this case, a cell from a mammary gland, into an egg cell that had its nucleus removed. The egg was then stimulated to develop into an embryo and implanted into a surrogate mother.
Cloning a mammal posed numerous scientific challenges. One of the most significant was reprogramming the adult cell's nucleus to revert to an embryonic state, allowing it to direct the development of a new organism. The process required precise timing and conditions, and previous attempts had failed to produce viable embryos. Another challenge was the ethical considerations of cloning a mammal, particularly regarding the potential applications of such technology in humans. The prospect of cloning raised questions about identity, individuality, and the moral implications of creating life in a laboratory setting. Despite these challenges, the team at the Roslin Institute pressed on, driven by the potential benefits of cloning for medicine, agriculture, and understanding developmental biology.
After many failed attempts, the team successfully cloned a sheep from an adult somatic cell. On July 5, 1996, Dolly was born to a surrogate mother, appearing as a normal, healthy lamb. However, it wasn't until February 22, 1997, that the world was introduced to Dolly through a groundbreaking announcement. Named after the singer Dolly Parton, as a nod to the source of the original cell, a mammary gland, Dolly became an instant global sensation. Her existence confirmed that it was possible to clone a mammal from an adult cell, proving that specialized cells could be reprogrammed to create an entirely new organism. Dolly was genetically identical to the sheep from which the original mammary cell was taken, effectively making her a clone. Dolly's birth was a scientific triumph, but it also sparked a wave of ethical and philosophical debates. Questions arose about the implications of cloning for humans, the potential for cloning endangered species, and the possibility of cloning organs for transplantation. The scientific community and the public grappled with the promises and perils of this new technology.
Dolly lived a relatively normal life at the Roslin Institute, giving birth to 6 lambs through natural mating, demonstrating that cloned animals could reproduce. However, Dolly's life was not without complications. In 2002, at the age of 5, she was diagnosed with arthritis, which led to concerns that cloned animals might age prematurely. In February 2003, Dolly was euthanized after being diagnosed with a progressive lung disease. She was 6 years old, about half the normal lifespan for a sheep. Dolly's death raised further questions about the long-term viability and health of cloned animals. Despite these concerns, the creation of Dolly had already revolutionized the field of genetics and biotechnology. Her existence opened the door to advancements in cloning, stem cell research, and regenerative medicine. Scientists have since cloned other animals, including cows, pigs, and dogs, and have made significant progress in understanding the mechanisms of development in cellular differentiation. Dolly's legacy extends beyond her life. She remains a symbol of scientific curiosity and the potential of biotechnology to change the world. The ethical discussions sparked by her creation continue to shape public policy and scientific research, guiding how cloning technology is used and regulated.
Dolly the sheep's birth was a watershed moment in the history of science, demonstrating the power of cloning and the possibilities it presents for medicine, agriculture, and understanding life itself. While her life and death raised important questions about the ethics and long-term effects of cloning, Dolly's existence remains a testament to the remarkable achievements of genetic science. As we reflect on Dolly's legacy, we are reminded of the delicate balance between scientific advancement and ethical responsibility in the pursuit of knowledge.